Today I'm going to be talking about how language relates to the brain. This is known as neurolinguistics. Neurolinguistics is an interdisciplinary field that pulls from neuroscience and linguistics, among others, but I'm going to focus on the biological and physiological aspects of it. It's essentially the study of how the brain comprehends, produces, and learns language. How do we study neurolinguistics? The technology used here is relevant because it's what helps us understand the structure and function of the brain. There are two types, hemodynamic and electrophysiological. Hemodynamic techniques, including the PET scan and fMRI, take advantage of the fact that when an area of the brain is at work, blood is sent to supply oxygen to that area. Since they can show which parts of the brain are active at a given time, they have high spatial resolution, but low temporal resolution because the bulb response is slow. Electrophysiological techniques take advantage of the electric currents of groups of neurons in the brain that fire together. EEG measures this electric current using sensors on the scalp, while MEG measures magnetic fields generated by these currents. And these have high temporal resolution, but low spatial resolution. Language is a defining characteristic of humans because other animal communication lacks a key aspect of human language, the creation of new patterns of signs under different circumstances. First language acquisition is particularly interesting because it's a phenomenon different from learning, so it's distinguished from second language acquisition. Noam Chomsky, known as the father of modern linguistics, rooted his linguistic theory in biolinguistics, as he said that the principles of the structure of language are biologically determined in the human mind and thus genetically transmitted. In humans, the nervous system is divided into two parts, central and peripheral. The central nervous system consists of the brain and spinal cord, which act as a control center. From there, cranial nerves and spinal nerves branch out to the rest of the body to form the peripheral nervous system. Motor nerves transmit information from the central nervous system to the body, while sensory nerves transmit information back to the central nervous system. Nerves are made up of axons of neurons, forming cylindrical bundles of fibers. The structure and function of neurons will be covered in detail next. A neuron has three main parts, the dendrites, cell body, and axon. The dendrites receive messages from other neurons, and the axon passes the message down and transmits it to other neurons. In order to understand how a neuron works, we have to understand the salty banana membrane structure. The diagram shows a section of an axon membrane with sodium ions outside and potassium ions inside. Although they both have a positive charge, there are more sodium ions than potassium ions, so the inside of the axon is relatively negative to the outside. When a signal is detected, sodium channels open to allow for an inward flow of sodium. This triggers channels further down to open, creating a cascade effect that moves in the direction away from the dendrites. After the inside charge increases to a maximum, potassium channels and sodium-potassium pumps restore the gradient to the original state. Here the graph of the action potential shows how the membrane voltage changes as a message passes down the axon. The membrane at its resting state is polarized. Charges greater than that are depolarized, while charges less than that are hyperpolarized. The threshold ensures that enough stimulus is present before the neuron carries out the signal. And the strength of the stimulus is correlated to the frequency of the action potentials, so more action potentials over a period of time indicate a stronger signal. Synapses are the connections between neurons that allow them to communicate messages. After the action potential travels down the length of the axon, it reaches the axon terminal and triggers the opening of calcium channels, allowing for an inward flow of calcium ions. Calcium then causes the release of neurotransmitters from vesicles into the gap between the presynaptic and postsynaptic cell. Neurotransmitters are chemicals that can be either excitatory or inhibitory, depending on the properties of the receptor. Excitatory neurotransmitters depolarize the postsynaptic membrane in order to reach the threshold for an action potential, while inhibitory neurotransmitters hyperpolarize the membrane in an effort to stop the action potential. Because a neuron has many dendrites for receiving neurotransmitters, the balance between the two types of neurotransmitters determines the charge of the membrane and whether an action potential is sent. Something that is central to learning and memory is plasticity. Neuroplasticity is the ability of the brain to change physically throughout a lifetime. More specifically, synaptic plasticity is the ability of synapses to become stronger or weaker in response to their activity levels. Hebbian theory describes a basic mechanism for this change, where the presynaptic cell's repeated stimulation of the postsynaptic cell leads to increased effectiveness of communicating the message, or in other words, increased synaptic strength. This is summarized as neurons that fire together wire together. When synaptic plasticity produces a long-term increase in signal transmission, it's called long-term potentiation. As a result, some physical changes include more neurotransmitter receptors or more neurotransmitters released. So how does this relate to language acquisition? A group of neurons connected by synapses makes up a neural network that is strengthened by learning. Young children have many more but weaker neural connections than adults because these connections either strengthen or disappear as people age. The critical period refers to the first few years of life when the nervous system is especially sensitive to environmental stimuli, such as the speech of parents. In fact, studies show that infants can respond to sound as early as 10 weeks before birth. An infant's brain responds best to a type of speech called parentese, which is what people use naturally when speaking to babies. This includes higher pitch, more inflection, and short, simple sentences. Other studies show that there is a sensitive period in which infants have the ability to learn any language, which is thought to be from birth to 6 months. After that, cooing and babbling are milestones in language acquisition because the baby is able to imitate sound patterns. The widely accepted critical period hypothesis states that the first few years is the crucial time in which a person can acquire a native language, and that if language input does not occur during this period, they will never achieve a full command of language. The brain has two hemispheres with different functions connected by the corpus callosum down the middle. Language is governed by the left hemisphere along with logic and math. The left hemisphere is dominant for those who are right-handed. This is a left side view of the brain. It's made up of 3 parts: the cerebrum, cerebellum, and brainstem. The cerebrum contains the cerebral cortex in which most of the higher functions, like thinking and processing information, occur. The cerebellum coordinates movements, and the brainstem serves to connect the brain to the spinal cord. The material in the cerebrum can be categorized into gray matter and white matter. Gray matter is made up of neuron somas, or neuron cell bodies, and it's called the cortex. It forms the outer layer of the cerebrum, or the cerebral cortex. The interior of the cerebrum mostly consists of white matter, or collections of axons. Also note that these wrinkles on the cortex help to increase surface area, which allows for more gray matter and more neurons. The cerebral cortex is further divided into 4 lobes. The most important takeaway from this diagram is that there are distinct parts of the brain that perform separate functions. We'll only be concerned about Broca's area in the frontal lobe and Wernicke's area in the temporal lobe. Aphasia is defined as a loss of ability to understand or express speech, which is caused by brain damage. Broca's area is in charge of speech production, which explains why it's near the motor cortex, because it has to coordinate tongue and lip movements. When this part of the brain is damaged, expressive aphasia results in lack of fluent speech, which means grammatical errors and slow or halting speech. However, they can still understand language, so the words they say are meaningful. The second type of aphasia is caused by damage to Wernicke's area, in the temporal lobe near the auditory cortex. This area is responsible for understanding the language that is heard. The information is then conveyed to Broca's area in order to produce a coherent response. Receptive aphasia means that the person can speak with ease, but their speech doesn't make sense to others. So you can see how both of these areas in the brain are necessary for us to produce and understand language. Going back to the idea of plasticity, the brain can modify itself physically by increasing or decreasing the proportion of gray matter in response to experiences like learning and memory. This can be observed in the well-known scientific study shown in the diagram. As a person gains knowledge, they strengthen neural networks, which also helps to organize the brain. In the same way, neural connections can gradually weaken from disuse. During early infancy, language processing seems to occur over many areas in the brain, but over time it becomes concentrated in Broca's area and Wernicke's area. Another interesting example is that the brains of bilinguals look and function differently than brains of monolinguals. While we still need more evidence for this, it definitely shows that language plays a big part in shaping our brains from the moment we're born. Even though neurolinguistics is a relatively young field, it's rapidly advancing with modern technology.